which will be relevant to the PG medical entrance as well as for the second year MBBS students. In general microbiology, they can ask various MCQs like identify the images of various eminent microbiologists. Okay. So, this particular figure is Antony van Leeuwenhoek. Can you tell me the discovery of this particular eminent microbiologist? He has discovered microscope, simple microscope. Louis Pasteur. Next one is Robert Koch. And last one is Paul Ehrlich. Of course, you will be knowing the various contribution of each of them. I am not going to go into the detail of this. Next is the differences between a gram positive and a gram negative bacterial cell wall, especially the peptidoglycan. When you look into the peptidoglycan layer, the peptidoglycan layer of gram positive cell wall is composed of alternate NAG and NAM molecules. NAG is N-acetylglucosamine, NAM is N-acetylmuramic acid. So, even gram positive or gram negative cell wall, both the peptidoglycan layer is composed of multiple layers, and each layer will have alternate NAM and NAG molecule. The difference is from each NAM molecule, in gram positive there arises a tetrapeptide side chain. Side chain, and both the tetrapeptide side chains are joined together by a pentaglycine bridge. And the tetrapeptide side chain of gram positive cell wall, the sequence is L-alanine, D-glutamine, L-lysine and D-alanine. The difference is in gram negative. So, this is gram positive cell wall. In gram negative cell wall, from NAM molecules the tetrapeptide side chain will be there, but the difference is at the third position, instead of lysine you have mesodiaminopimetic acid. So, this is the difference, one difference. Second difference is both the tetrapeptides are directly linked to each other, that is there is no pentaglycine bridge. Here both the tetrapeptides are joined by a pentaglycine bridge. Here there is no pentaglycine bridge, there is directly both the tetrapeptides are joined to each other. So, these are the main difference between gram positive peptidoglycan and gram negative peptidoglycan. Coming to the important identifying culture medium, this is the peptone water broth, first one, peptone water broth, which is used as a which type of medium? Basal medium. The second one is a nutrient agar, this is also a basal medium. This is blood agar and this is chocolate agar. Chocolate agar is heated blood agar, where the blood is poured at 70 to 75 degree centigrade. The blood is poured at this temperature to the medium, that is why the blood is lysed and chocolate agar is formed. So, these are other selective medium, this is LJ medium, Lowenstein-Jensen medium, and this is TCBS agar centigrade. The blood is poured at this temperature to the medium, that is why the blood is lysed and chocolate agar is formed. So, these are other selective medium, this is LJ medium, Lowenstein-Jensen medium, and this is TCBS agar. LJ medium is used for mycobacterium, tuberculosis. TCBS agar is used for Vibrio cholera, and these two are blood culture bottles. Blood culture bottles composed of, this is a biphasic medium, this is a monophagic medium, whereas this one is a biphasic medium. Monophagic medium is composed of brain heart infusion broth, whereas biphasic medium is composed of brain heart infusion agar and broth, both. Okay, so blood culture bottle, this is used for culture of blood sample. This is antibiotic susceptibility test. This medium is called as Muller-Hinton agar, MHA, Muller-Hinton agar, and this particular method is called as disc diffusion test. Kirby Bowers disc diffusion test, and in this test they say that if you will be doing, first you will be doing a lawn culture of the organism. The organism is, you have to do a lawn culture or the carpet culture of the organism, you have to do over the culture medium. After that you have to place the disc. If there is growth surrounding the disc, then this is considered as resistant. If there is no bacterial growth surrounding the disc, this is considered as organism is sensitive to the antibiotic. And this is a higher up, this is a modern variety of antimicrobial susceptibility test, which is called as E-test. This is also a type of antimicrobial susceptibility test called as E-test, and E-test is used for calculating the MIC. MIC is a minimum inhibitory concentration. So MIC of the particular antibiotic is calculated by E-test. The full form of E is epsilometer test. Okay, it works on the principle of both diffusion and dilution, whereas here only diffusion principle is used, whereas here both diffusion and dilution principle is used. So let us now concentrate on images of systematic bacteriology. So coming to the images of Staphylococcus aureus. Staphylococcus aureus is catalase positive. Catalase positive means when you add 3% hydrogen peroxide, bubbles will appear, then it is considered as catalase positive. Gram stain, gram positive cocci in cluster. Nutrient agar Staph aureus produces golden yellow colonies. Golden yellow colonies are made of beta carotene. Blood agar Staph aureus produces pinhead colony with what kind of hemolysis? Narrow hemolysis, narrow zone of beta hemolysis, beta or complete hemolysis. This is mannitol salt agar. This is a selective medium of Staph aureus. Selective medium of Staph aureus. Coagulase test, tube coagulase test. This is, they say that the tube coagulase test, if it is positive, then the clot will be formed. Clot will be formed. If it is negative, then the medium looks, it will be turbid, but there is no clot formation. If the tube coagulase, if it is positive, then on tilting there will be a clot formation. Example, the tube coagulase and slide coagulase both are positive for Staph aureus, Staph hyacus and Staph intermedius, whereas tube coagulase negative, slide coagulase positive. Example is Staphylococcus lugdunensis. Lugdunensis. Slide coagulase negative, tube coagulase positive. Example Staphylococcus scleferi. Okay. This is a catalase test. Again, it is a repeat slide. It is a catalase test which we have already discussed. Okay. So, in general, we will summarize all the figures of Staphylococcus aureus. So, the figures are gram positive cocci in cluster, nutrient agar golden yellow colony, blood agar pinhead colony with beta hemolysis, narrow zone, mannitol salt agar, and this is tube coagulase positive, and yes, this is the figure of slide coagulase. So, slide coagulase test, if it is positive, then clumps will be produced, and slide coagulase, I hope you know, it is due to clumping factor, whereas tube coagulase is due to coagulase enzyme. Okay. So, this in this figure, catalase is positive, tube coagulase is negative. This is a novobiosine disc, and in this figure it is shown that this organism is resistant to novobiosine. This is typically it is a case of Staphylococcus saprophyticus, which is resistant to novobiosine, and it is a coagulase negative staph, that is why tube coagulase is negative. Coming to the figures of Streptococcus. Streptococcus can produce various clinical manifestations like impetigo and erysipelas. Erysipelas will have a pudi orange skin, mainly on the malar area. Erysipelas is a variant of cellulitis. Impetigo is characterized by honey color crust. Okay. This is necrotizing fasciitis. Necrotizing fasciitis. And Streptococcus will be catalase negative. All the Streptococci will be catalase negative. Coming to Streptococcus pyogenes, gram stain will be gram positive cocci in chain. I have also kept the Staphylococcus gram stain so that you can easily differentiate. Staph will be gram positive cocci in cluster, whereas Stret will be gram positive cocci in chain. So, that is how the pictures will be differentiated. Similarly, Streptococcus pyogenes, blood agar pinpoint colony surrounded by wide beta hemolysis. This has to be differentiated from that of Staph aureus, which is pinhead colony surrounded by narrow hemolysis. And group B Streptococcus will produce a positive CAMP test. That is, in a blood agar plate, center you streak Staph aureus streak line, and perpendicular to that you streak group B Streptococcus. Both Staph aureus and group B streptococcus are beta hemolytic. You will have a yellowish zone of discoloration on both the streak line. But the difference is the group B streptococcus will produce CAMP factor. This CAMP factor will be produced by group B streptococcus, and this will synergistically enhance the hemolysis at the junction of Staph aureus. Group B streptococcus will produce CAMP factor that will synergistically enhance the hemolysis at the junction of Staph aureus. So, this is called as a positive CAMP test, and this is done for group B streptococcus. Coming to the features of Enterococcus. Enterococcus is gram positive cocci in pair, spectacle eye appearance. Spectacle eye appearance. Blood agar you will have non-hemolytic colony, non-hemolytic colony, and here you have a look. This is a positive bile esculin hydrolysis test, and this will be the this is a negative control. So, positive bile esculin hydrolysis test is a feature of Enterococcus. Okay. Viridans streptococcus will be alpha hemolytic, green hemolysis, and of course gram positive cocci in long chain. This has to be differentiated from Pneumococcus. Pneumococcus gram stain will be gram positive cocci in pair, lanceolate sept, like this, lanceolate sept or flame sept surrounded by a capsule, surrounded by a capsule. And on blood agar, alpha hemolytic colony, but the colonies are bigger in size, having a dimple. It's called as Draughtsman sept or carom coin colony. Carom coin colony. And on chocolate agar and pneumococcus will produce bleaching effect. Bleaching effect on chocolate agar. So, this is Draughtsman sept colony or carom coin colony, and this is the bleaching effect of pneumococcus on chocolate agar. Of course, pneumococcus will be soluble in bile, that is bile will become clear when you inoculate with pneumococcus, in contrast to viridans where the bile will be turbid, that is insoluble. When you keep a optochin disc at the center, you will have a big zone of sensitivity. Why? Because pneumococcus is sensitive to optochin. Okay. Then coming to Neisseria meningitidis, gram negative cocci in pair, lens sept or half moon sept. Neisseria gonorrhoeae, gram negative cocci in pair, kidney sept. Of course, intracellular, both are intracellular, both are intracellular. And the previous one, meningococcus, is capsulated, gonococcus is non-capsulated. Corynebacterium, this is having a, so Corynebacterium broadly two type, Corynebacterium diphthery and others are called as diphtheroids. Diphthery will have a Chinese letter arrangement, having metachromatic granule. So, this actually this picture is Albert Stein picture. Then do Albert Stein, metachromatic granules will appear as bluish black metachromatic granule and green bacilli, and they are arranged in Chinese letter or cuneiform arrangement. In contrast, diphtheroids, they are palisade or parallel to each other, palisade in arrangement. Of course, this is the gram stain, these are gram positive bacilli in palisade arrangement. This is Loeffler's serum slope, it is an enriched medium for Corynebacterium diphthery, and this is the selective medium for Corynebacterium diphthery, that is potassium tellurite agar, where Corynebacterium diphtheria produces black colony. And these are the manifestation: membrane over the tonsil, pseudomembrane over the tonsil, and this is the complication seen in diphtheria called as bull neck. Bull neck is neck edema plus tonsillar swelling. Neck edema plus tonsillar swelling. Bacillus anthracis. Next, this particular picture is, it denotes malignant pustule. Is it malignant or not? It has no malignancy. Because of the angry look, it is called as malignant pustule. Gram stain appearance, gram positive bacilli, long chain. Which appearance? Bamboo stick appearance. Bamboo stick appearance. McFadden reaction. Polychrome, methylene blue stain. Okay. So, blue bacilli surrounded by amorphous purple capsule. Blue bacilli surrounded by amorphous purple capsule of Bacillus anthracis. This is a nutrient agar under microscope. The colony will appear as Medusa head colony. Medusa head colony. Anthrax on blood agar, non-hemolytic dry wrinkle colony, and on gelatin stab agar, inverted fir tree appearance colony. Inverted fir tree appearance colony. Okay. Next is Clostridium. There are various arrangement of Clostridial spores. Most of the Clostridium, the spore arrangement is subterminal. This is the most common form. Terminal and round give rise to drumstick appearance seen in Clostridium tetani. Terminal and oval giving rise to tennis racket appearance. Clostridium tertium. And central spore, example is Clostridium bifermentans. Clostridium bifermentans. Then Robertson cooked meat broth is the most common anaerobic medium. This is an uninoculated medium. They say that Clostridium perfringens being saccharolytic, it will turn the color of the meat to pink. In contrast, Clostridium tetani being proteolytic, it will turn the color of the meat black and having a very putrid smell, offensive smell. Then Clostridium perfringens manifestation, this particular picture is a clinical case of gas gangrene, presence of bubbles in the muscle compartment. And when you do a gram stain, if you get gram positive bacilli without spore having a typical box car shaped appearance, this give rise to the clinical suspicion that it could be Clostridium perfringens, because the gas gangrene strains of Clostridium perfringens are non-sporing, they are non-sporing. And further confirmation can be done by performing a reverse CAMP test. Clostridium perfringens confirmation can be done by performing a reverse CAMP test, by observing target hemolysis or double zone hemolysis, and the most important is positive Nagler reaction. Yeah, here it is there, a positive Nagler reaction. Nagler reaction is nothing but you will have a opalescence on egg yolk agar. Opalescence on egg yolk agar, which is due to alpha toxin and which can be inhibited by adding anti alpha toxin. Clostridium tetani various clinical manifestation, opisthotonous position, trismus and rhizus sardonicus, grimming movement of the facial muscles, rhizus sardonicus, drumstick appearance spore, characteristic feature of Clostridium tetani, swarming on blood agar. Clostridium tetani typically it swarms on blood agar. This is pseudomembranous colitis. Agent is Clostridium difficile. So, these are the pseudomembranes on the colonic mucosa. Colonic mucosa, presence of pseudomembrane, and this is the histopathological feature. Actinomyces again is gram positive, branching gram positive bacilli, branching gram positive bacilli, and they say that actinomyces can cause cervicofacial lesions, cervicofacial lesions. And this is nocardia. Nocardia is acid fast branching acid fast bacillus. Okay, partially acid fast, partially acid fast. Mycobacterium tuberculosis, this is the typical appearance on Jaden stain, that is long, slender, beaded appearance. And the next picture is auramine phenol stain, fluorescent stain, auramine phenol stain or auramine rhodamine stain. The mycobacterium appears as fluoros, brilliant green, fluorescent color. Algae medium, the description is rough, tough and buff color colony. Mycobacterium lepra in bundles, cigar like bundles of acid fast bacillus, cigar like bundles of acid fast bacillus arranged in globi. All these are globi actually, arranged in globi. Okay, so cigar like bundles of acid fast bacillus arranged in globi. The globi are present inside a lipid macrophage, and the lipid laden macrophage is called as Virchow's lepra cell. Here of course the macrophage outline is not clear, but globi, the clumps of bacilli are clearly seen. Okay, so these are the various lesions produced in leprosy. This is a case of lepromatous leprosy, whereas this is anesthetic lesion of tuberculate leprosy. The descriptions you can read from book. These are the various deformities seen in leprosy. Okay, corneal opacity, okay, nose deformity, atrophy of digits, finger deformities, all these are seen in leprosy. Okay. Now, I do not know whether it is how much this is useful, because till now in the image based exam they have not asked the interpretation of biochemical reactions, but at least broadly I will outline the 3-4 important organisms like E. coli, Klebsiella, so that even if it is asked you should be in a position to answer. Okay. So, catalase test I have already told you, if you add 3% of hydrogen peroxide, then effervescence will be there, bubbles will be there if it is positive, and all enterobacteriaceae catalase is positive. All enterobacteriaceae are catalase positive. Can you tell any exception? Shigella dysentery type 1, which is catalase negative. Okay. Oxidase, if the test is oxidase positive, then the paper turns blue, the filter paper turns blue. Okay. Indole test, they say that if the test is positive, a cherry red color ring will be produced, cherry red color ring will be produced. If it is negative, then no ring will be produced. I mean a colorless ring, colorless or the color of the medium, whatever ring will be produced. Citrate, the original color of the medium is green, green is the original color of the medium. If positive, it turns blue. Urease, the original color of the medium is pale, if positive, it turns pink. This is very difficult to understand. This medium is called as TSI, triple sugar triple sugar ion test. Okay, triple sugar ion test. They say that this is the picture of an uninocculated medium. Here why it is called as triple sugar ion test, because here 3 sugars are there: glucose, sucrose and lactose. Glucose 1 part, whereas sucrose and lactose are 10 parts more in amount. Okay. So, this is a uninoculated medium. First the picture is an uninoculated medium. It has a slant, it has a butt. Both the slant and butt are pink in color. The principle of this test is, if the organism is growing in this medium, fermenting some sugars, then sugar fermentation leads to a production of acid, and acid production makes the color yellow. If sugar is not produced, then the pink color or the alkalinity will be maintained. If no sugar is fermented, then the pink color or the alkalinity is maintained. So, one property you want to test here, whether the organism is fermenting sugars or not. If fermenting sugars, whether all 3 sugars are fermented or only glucose is fermented. Second property you are going to test here, gas is producing or not. If the organism is producing gas, then you will have, the medium will be lifted. The medium either will be lifted or maybe the medium will be broken into pieces. Break will be there in the medium. Third property is whether the organism is producing H2S or not. If H2S is produced, then the medium will become black. See here, the medium will become black. Fine. If you are done with this, I will tell you now which organism will produce what kind of reaction. The commonly isolated gram negative organisms, Enterobacteriaceae organisms, are broadly categorized into non-fermenter. Example is Pseudomonas, glucose only fermenter. Example is Shigella, Salmonella, Proteus, etc. Glucose, I mean all fermented. The third group is all fermenter. All fermenter is E. coli and Klebsiella. Clear? Non-fermenter group do not produce any acid because they do not ferment any sugar. So, the alkalinity is maintained. That means slant is alkaline, but is alkaline. Okay, we give a code of K for alkaline. So, K by K is the code, and Pseudomonas do not produce gas, do not produce H2S. So, K by A, gas minus H2S minus is the result of Pseudomonas. All fermenter like E. coli and Klebsiella, when all these 3 sugars are fermented, enormous acid will be produced. So, the entire medium will become acidic or yellowish. Slant will become acidic, but also will become acidic, and we give a code of A for acidic. So, it is A by A, and both E. coli and Klebsiella, they produce gas. So, B is the a picture of both Klebsiella and E. coli. Both Klebsiella and E. coli, they produce gas and they do not produce H2S. They are gas plus but H2S minus. Clear? Then comes glucose only fermenter. So, glucose only fermenter, glucose is more in amount or less in amount? They are very less in amount. So, they say that only the butt will be acidic, like this, only the butt will be yellowish, slant will be alkaline, because only glucose is fermented. So, the reaction will be K by A, reacts all Shigella, Salmonella, Proteus, everywhere the reaction will be K by A. In Shigella, K by A gas minus H2S minus, that is number C. Number C is for Shigella, gas minus H2S minus. Proteus, K by A gas minus but H2S plus, that is number E. This is Proteus. Remember, Proteus H2S production is so much that the entire butt will look black. So, inside the yellowish color will be there, which will not be look to you, only the outer black color will be seen. In Salmonella typhi, of course K by A, there is no gas, gas will be minus, H2S will be tiny, T for typhi, T for tiny. Tiny means only little bit H2S at the junction. Which one can you guess? D. Little bit of H2S will be there in the junction. This is Salmonella typhi, this is Shigella. Clear? Clear? Okay. Identifying a feature of common gram negative bacilli. E. coli. E. coli on